I'll be talking today about work that, as I mentioned, was done in coordination, collaboration, and in support of the US EPA's Endocrine Disruptor Screening Program. And it's all about pathway-based models for rapid screening of endocrine disruptors. So you've already heard about NICEATM and IVIVE, so I won't spend any time on that. I also won't belabor the point of what TOX21 is, because I know that everyone in the room is familiar with that, and the concept of how we're going from high throughput screening to pathways and pathway-based models that use computational toxicology approaches to try and both in a qualitative fashion prioritize chemicals for targeted testing and hypothesize possible mechanisms, but also more and more in a quantitative fashion to actually screen chemicals for hazard. And then with some of our friends from industry, from a bottom-up type approach, actually design chemicals that minimize hazard while still maximizing their efficacy. So environmental endocrine disruptors have been a concern for many decades. Back in the early 90s, Theo Colburn and colleagues published a seminal paper observing that there were developmental effects of endocrine disrupting chemicals in the environment and that these could potentially translate into humans. As a result of this work and many other pieces of work in the literature, there were some legislative mandates that were passed in the mid-90s. The Food, Drug, and Cosmetics Act and the Safe Drinking Water Act led to the formation of the US EPA Endocrine Disruptor Screening Program. So the Endocrine Disruptor Screening Program, or the EDSP for short, is as I mentioned, a congressionally mandated screening and testing program that has two tiers. So tier 1 testing, and that's what we'll be focused on today, is for the purposes of prioritization and screening. We want to identify chemicals that have the potential, and that's an important word, a potential to disrupt estrogen, androgen, or thyroid hormone receptor signaling. Tier 2 is more focused on whole animal and adverse outcome events. As I mentioned, Tier 1 is more about prioritization and screening. But Tier 1 already in and of itself is incredibly resource and time intensive. So it costs about $1 million per chemical to screen a chemical through the 11 low throughput and animal tests that make up Tier 1 testing. And so you can see here, this is the EDSP chemical universe. So there's about 10,000 chemicals under FIFRA and CIDWA. The first set of chemicals where EPA issued test orders was about 67. Second list of chemicals where test orders actually have not been issued yet, because we're reevaluating based on some of the work I'll tell you about today. It's about 100 chemicals, so really drops in this large bucket. But we, through the ToxCast and TOX21 program, have generated high throughput screening data on over 3,000 chemicals. So you can see here that actually starts to make a big difference. That's like a big splash in this bucket. And so this approach, the EDSP approach to screening for endocrine disruptors, plus the TOX21 high throughput screening approach, is being referred to as EDSP21. And so what we've been doing is to try and build predictive models that are based on our knowledge of the biological pathways in question here. Specifically, I'll be talking today about the nuclear hormone receptors, the estrogen and the androgen receptors. These involve multiple high throughput screening assays that are orthogonal assays, providing different and complementary pieces of information along the biological pathway. And then we ask the question of whether or not these high throughput screening based pathway models were sufficient to replace selected tier 1 screening assays. So how do we validate models to replace other models? Because ultimately an animal is simply just a model of a human being. So in the case of validation, I always turn to what I refer to affectionately as the Bible of validation. This is OECD Guidance Document 34. And GD 34 states that validation is a process by which the reliability and relevance of a test method are established for a specific purpose. And so we've already established what the purpose of EDSP Tier 1 screening is. We're trying to identify chemicals that have the potential to be endocrine disruptors via these nuclear hormone receptor pathways. And validation is also performance-based. And so how do you evaluate the performance of a new test method? To do so, we rely very heavily on reference chemicals. So these are chemicals that we understand, that we know something about, that they've been tested in the original test method that's currently accepted by regulatory agencies. Ideally, this is a wide range of chemicals with varying potencies and chemical structures and chemical use cases. And then we can use these reference chemicals to compare the results of our new test system or our new model against the existing system. So the work I'm going to start off telling you about is not new by any means. It was published at the end of 2015, so it's been out in the literature for about a year and a half. There's still some misconceptions floating around about the work, so hopefully even those of you who are familiar with it will find this clarifying, and those of you who aren't, hopefully you'll find it interesting. And this work forms the basis of ongoing work both in the science and the science policy space that I'll talk to you about towards the end. And so these are the three publications that I'm going to cover at a very high level. So in the ToxCast and Tox21 programs, we have many different assays that map to the estrogen receptor signaling pathway. So these are binding assays, alpha beta, and beta beta dimerization assays, DNA binding, RNA transcription, protein production, all the way down to ER-controlled cell proliferation. These come in a variety of systems ranging from cell-free to different cell types. Most of them are human. We do have a mouse and a bovine binding assay in addition to the human binding assay, and they're offered by a variety of vendors. And the vast majority of these assays were provided via the EPA's ToxCast program. Some of them, the ones highlighted in blue down here, are from the NIH, and that should be NCAT's, National Center for Advancing Translational Science, under the Tox21 program. So how do we combine the results from these multiple assays, and why do we combine the results from these assays? Why don't we just rely on a binding assay by itself or a transactivation assay? Well, that's because no individual assay is perfect. Every test system has specific flaws that relate to the technology that's being used, potential human error. There's all kinds of different sources of noise and sources of error that can be introduced into different technologies. And so we leverage the orthogonality of having different assays that measure both the same and complementary points along a biological pathway, and we integrate all of those assays together in a weight of evidence approach that provides us an indication of whether or not a chemical is actually acting as, for example, an estrogenic chemical via this nuclear hormone receptor. And we use a pretty simple mathematical model to integrate this data together. It's very important to acknowledge the fact that this is not a machine learning model. There is no training data here. This, if anything, the only way the model is trained is based on our biological understanding of the pathway. So the concept that a chemical that's going to be acting through this pathway first would bind to the receptor, and then it would dimerize, and then it would go through DNA and RNA transcription all the way down to protein proliferation and cell proliferation. And so that biological understanding is what drives the formation of this very simple constrained least squares regression model. And the only quote unquote training chemical in the model would be our positive control chemical. So this is 17-beta estradiol. And so we can integrate the dose response curves from all of these different assays into one composite dose response curve for the pathway. And then we can compare that to our positive control estradiol and calculate an area under the curve and compare that to and normalize it to estradiol. So here's some example curves. So everybody's favorite chemical, BPA, these are all the individual concentration response curves from all the in vitro assays. This blue curve right here is the integrated synthetic concentration response curve for the pathway. It has an AUC value of 0.65. That's respective to estradiol, which has an AUC value of 1. So everything's normalized to the positive control here. You see some little blips here, which these are interference pathways. So the model initially starts, so these assays right here are more sensitive and they become active at lower concentrations. So initially the mathematical model thinks like, oh, well, maybe this is interference, but then all these other assays further downstream in the pathway turn on. And the model says, no, actually, this is true ER pathway activity. And so this is the composite concentration response curve. This is the same thing for a true antagonist. So here the red is the concentration response curve for an antagonist. So raloxifene hydrochloride is also a positive control compound for ER antagonism. And you can see it doesn't really look like a hill curve because it's maxing out at basically the lowest tested concentrations. And then alpha cyclodextrin is a good example of a compound that's acting through assay interference. So in a couple of the assays, it's interfering with the technology, and it's not actually acting as a true ER pathway agonist or antagonist, and the model correctly predicts that. So this is the composite dose response curve for an interference pathway in the model. So to evaluate the performance of the model, we looked at reference chemicals. So there are a number of reference chemicals in the literature for the ER pathway. Most of them are in vitro reference chemicals. So reference chemicals that have been very well tested in a number of different in vitro assays and a number of different platforms. And so you can see here that the red are the active, the blue are the inactive. This is the AUC score for the model, ranging from your positive control all the way down to negative. And we get very high accuracy. We get 95% accuracy against these in vitro reference chemicals. But we were more interested in the in vivo scenario. So the EDSP Tier 1 guideline test for screening for estrogenic activity is the rodent uterotrophic bioassay. So this animal test uses a couple different animal models that are acceptable. You can use an overrectomized rat or an overrectomized mouse, or you can use a sexually immature rat. The key here being that the animal is not producing its own endogenous estrogens, and instead you dose with exogenous estrogens, and you see whether or not you get a response in uterine cell proliferation that is measured as an increase in uterine weight. And so we went through the literature and tried to identify all of the uterotrophic studies that had ever been performed and published in the scientific literature. And then we evaluated them for their quality. So we looked at the OECD and the EPA test guidelines for how to correctly run a rodent uterotrophic study. And we came up with these minimum criteria. And these minimum criteria include using the correct animal model, having high enough statistical power so you have large enough group size in all of your dose groups, having the right controls, having the right routes of administration. So all of these minimum criteria that would make a published uterotrophic study equivalent to a guideline uterotrophic study that was run via the EPA or the OECD test guideline. And so from those high quality guideline like studies, and I should mention that this is a project that took over 2 years and pretty much every person in the NICEATM group touched this project at one point or another. We reviewed over 700 studies in the literature and had 2 people independently extract every piece of study protocol information, chemical information, and response information from all of these studies, and then narrowed down to the set of high quality guideline like studies, which was around 120 out of 700. So, less than 20% of what's in the literature is really guideline-like. Then from those high quality guideline-like studies, we identified a set of reference chemicals that we call in vivo reference chemicals because they have reproducible results in the high quality guideline-like studies. So they had to be verified in more than 2 independent guideline-like studies, and negative chemicals had to show no active results in any study. So when we narrow down to a set of extremely high quality studies, data that we have high confidence in, chemicals that have reproducible results in those studies, we get very, very high accuracy. So we get 97% accuracy against this fairly large set of reference chemicals. And I should mention that the rodent uterotrophic assay that was accepted as this, tier 1 estrogenic screening test for EPA was validated at the OECD using 7 chemicals. So we feel like we really went above and beyond in using over 80 chemicals to validate this. And this gives you the same picture that I showed before, but for the in vivo reference chemicals, you can see that we have good coverage in terms of different potencies. So of active and inactive chemicals, we have 1 false negative and 1 false positive. So our false negative is camphorol, and this is something that it was inactive in vivo, but it was active in vitro, and this is actually an in vitro reference chemical. So it's well known to be estrogenic in vitro, and then it's transformed to a inactive form in vivo. And then D4 was our only false negative. And so it was inactive in the in vitro assays, and it was active in vivo in 2 different studies, high quality studies. And this is a highly volatile chemical. And so this is a caveat that you have to take into account when looking at in vitro, specifically high throughput screening assays. One nice thing about the Tox21 program is that we have chemical QC results for all the chemicals that have been tested in the program. Some of them are still underway, but everything's being generated. And we were able to go look at D4, and we found out that this chemical had actually failed the chemical QC and very likely wasn't in the well when it was being tested. So interpreting the results in the context of the chemical QC is really important. So 2 years ago now, this work was presented and reviewed and accepted by a scientific advisory panel. And as a result, the EPA published a Federal Register notice announcing their intent to use high throughput screening assays, and specifically the ER pathway model, as an alternative to 3 of the low throughput EDSP tier 1 assay battery, including, importantly, the rodent uterotrophic assay. And this is really nothing short of historic. It's the first time that a US regulatory agency has ever accepted across the board a in vitro computational model, in vitro based computational model as a replacement for an animal test. So then we moved into the next pathway, which is the androgen receptor pathway. So this paper was just published in December, and I'll go through it pretty quickly because it's very, very similar to the ER pathway model and was in fact based entirely upon that work, but there are some key differences. So while the environmentally relevant activity of concern for the estrogen receptor pathway seems to be agonism, so we had around 270, 300 or so chemicals out of the 3,000 or so that have already been tested that appear to be estrogenic chemicals. Very few seem to be acting as antagonists. The opposite is true for the androgen receptor pathway. So the environmentally relevant activity of concern for androgen receptor is antagonism. We get very few chemicals that seem to be acting as androgenic or androgen receptor agonists, but we get many, many more that seem to be acting as anti-androgenic or antagonists of the pathway. So we have also good coverage of the AR pathway, not quite as many assays as ER, but we still have multiple binding assays, cofactor recruitment, RNA transcription and protein production, and then we have these antagonist assays. And as I mentioned, because this is the activity of concern, and antagonism is notoriously difficult to differentiate from cytotoxicity or generalized cell stress, we had to treat this model a little bit differently. So the reason antagonism is difficult to differentiate from things like generalized cell stress and cytotoxicity is that you're usually relying on loss of signal assays. And so to indicate an antagonist behavior. So you prep the assay with a known agonist, and then you look for competitive inhibition of the response based on treatment with an environmental chemical that might be an antagonist. So you're looking at this loss of signal, where that loss of signal could also be caused by unhappy cells or by cytotoxicity. And so what we did here is we ran one of the Tox21 assays, this antagonist assay, twice. And we used 2 different concentrations of the stimulatory ligand. And so what that gives us is 2 different concentration response curves. So we tested the environmental chemicals at the same concentrations, but we stimulated with different concentrations of the ligand. And so for a true antagonist that's acting through the receptor, you would expect to see a shift in that dose response curve based on the concentration of stimulatory ligand that you used in the assay. If you do not see a shift in the concentration response curve, and those curves are right on top of each other, that's likely a false signal that's not acting through the receptor. So that was an additional piece of information that we included in the AR pathway model. The literature around reference chemicals for the AR pathway is nowhere near as well developed as the ER pathway, so we embarked on another literature review. We actually did a targeted literature review in this case rather than just casting a broad net for anything AR. We started with the superset of chemicals that had all been ever considered as in vitro reference chemicals from various validation studies from different international organizations that are listed here. We found over 4,500 chemical study pairs in almost 400 papers, and then we went through the same thing where we extracted all of the information that we could, and this is just a exhaustive list of all of the information that was extracted from every paper. And then we looked at that information for its reproducibility across different labs, looked for chemicals that had consistent results that were independently reproduced. And I should mention that all of this, this database that we pulled from the literature and all of the information that was extracted from all the papers is all included in the supplemental material to the AR pathway model manuscript. So we came up with 29 agonist reference chemicals. We had 37, but only 29 of them were actually tested in the ToxCast and Tox21 assays, and 28 antagonists. And again, these are chemicals that have been tested in a wide variety of in vitro binding and transactivation assays and show reproducible results. And again, when we focus in on the high quality data and use that as our reference, we find that we get very high accuracy, 95 and 97% accuracy in predicting these reference chemicals. And this just shows you another screenshot, another way of looking at them. And the nice thing about this is that these are stratified by their outcome in the literature. So their potency, as far as the literature-based results, ranges from strong, and I should really, as I mentioned, there's not too many agonists. We've got lots of negatives in the agonist reference chemicals, but for the antagonists, we get a nice range of potencies. In some cases, you know, we didn't want to draw a hard line and say this is moderate, this is weak. So sometimes if their 95% confidence intervals spanned the boundaries of what we were calling moderate and weak, then we just gave them both designations. And you can see here that the AR pathway model score, which ranges from, this should be 0 because anything below 0.01 gets truncated to 0, and 1 tracks really well with the potency in the literature. And then the one compound that was a false negative, the AR pathway model predicted it as acting through assay interference because it didn't hit a couple of the upstream assays and it only hit the downstream assays. This compound, zearalenone, was actually identified by the confirmation assay results. And so when we put those confirmation assay results in tandem with the AR pathway model, we actually had 100% accuracy against the antagonist reference chemicals. So we're doing a similar thing as we did with the uterotrophic assay in doing a systematic literature review of the published Hirschberger experiments. So this is another rodent assay that's used to screen for androgenic and anti-androgenic activity. I will say that this is a much more complicated test than the uterotrophic. They're measuring 5 accessory sex tissues, and the protocols are not really as well defined. We have found so far that out of the almost 50 chemicals that we found with multiple studies in the literature, more than 80% of them have conflicting results, have both positive and negative results. And so now we're going through the process of looking for the consistency of the Hirschberger with other in vivo tests. So that's like the multi-generational or the male pubertal assays. And this is work that I'm doing in coordination with Patience Brown, who used to be at the EPA and is now at the OECD and lives in Paris, and we can all be jealous of her. So this work will hopefully be done within the next couple months. A couple of things I'll talk about quickly at the end of the talk is some of the explorative work that we've done in terms of uncertainty propagation. So looking at the uncertainty in the chemical assay responses and then propagating that through the pathway model. Some work that Kamel Mansouri has led in QSAR modeling to actually take the ER and the AR pathway results as training data and then use that to predict the entire EDSP chemical universe. Some IVIVE work to help understand how in vitro concentrations and activity levels actually translate to doses, and then some access to how do we access this data. So this is work that was led by Eric Watt, a former postdoc of Richard Judson, who now works at Pfizer, and he used a Bootstrap approach to look at all the different replicates of each chemical in each assay, and then put 95% confidence intervals around those chemical assay hit calls and fit values, AC50 values, and then we propagated all of those uncertainty bounds through the pathway models and created 95% confidence intervals around the model scores. So you can see here, this black line is drawn at 0.1, which is a model score that basically translates to activity against the pathway at 100 micromolar or below, so a score of 0.1 or above. And you can see here that some of the chemicals, even though their point values may be below that threshold cutoff of 0.1, where we're pretty confident that they're acting against the pathway, the upper bound of their 95% confidence interval, when you propagate the uncertainty that is derived from the different technical replicates of that chemical through the model, actually goes above that threshold. And so this is really helpful for regulatory prioritization and making sure that we're being conservative enough and inclusive enough in prioritizing our chemicals. So the QSAR Modeling project is a global collaborative project. So the first one was called SERAP. It was to predict ER pathway activity. There were 17 groups that participated. Kamel led this project, and it was published in EHP last year. And the prediction set is, as I mentioned, the training set was the ER pathway model for almost 1,700 structures, and then all the groups were challenged to predict out onto the EDSP universe of over 30,000 structures. And we're also doing the same thing for AR. So one nice thing is that when you look at the concordance of all the models, so each of the models, most of them I would say were pretty similar in terms of their performance, but each had strengths and weaknesses and certain areas of the chemical universe where they did better or did more poorly. But when the nice thing about a global challenge, is that you have many models that you can pull together and you can create a consensus model, and you can look at how many times those models all agree. And so we narrow it down to about 750 chemicals that have over 75% concordance among the different models. And so out of this vast array of chemicals in the universe, only a small fraction of them are actually prioritized for further testing. And this is being applied immediately in regulatory use by the Endocrine Disruptor Screening Program. And here is the EDSP dashboard, which is something that the EPA, National Center for Computational Toxicology, has put out. And you can actually find not only the ToxCast ER pathway and AR pathway model scores, but you can find the results of the QSAR modeling. For now, just the SERAP project, but soon also the Compara project for the AR predictions on this dashboard. Okay, so then the next thing I'll talk about very briefly is this concept of IVIVE. So you can look at an in vitro based model and you can say, oh, okay, I see that this compound is estrogenic at, say, 20 micromolar, but what does that actually mean in terms of real world exposures? How much of that compound would I actually have to eat to achieve plasma level or tissue level concentrations where I'm starting to see activity in my in vitro based model? And so that's what IVIVE does. We use reverse toxicokinetics and PK and PBPK, so physiologically based pharmacokinetic models, to actually do that translation from in vitro concentrations to predicted in vivo doses. And NICEATM and many, many other people, including people in this room, are working on developing open source tools to facilitate IVIVE. So we're working on an open source workflow in NIME, which you've seen multiple times in the last 2 days as this open source sort of pipeline tool. And here we get data. We use data from the in vitro ToxCast assays. We want to compare it to high quality in vivo data. Some key parameters here are hepatic clearance and plasma protein binding, and ideally these would be experimentally derived parameters, but we only have that for a few hundred chemicals, especially in the case of clearance. And so we and others at the NCCT are working on developing QSAR models to predict those parameters. So those will be, our QSAR models will be part of our open source workflow. And then you get an output of an estimated equivalent administered dose, and then you can compare that with the doses from your high quality animal studies. And this is just an example from a paper that's under submission at EHP right now, where we applied this workflow to compare estrogenic activity in the ER pathway assays. So these box plots are the equivalent doses estimated from the AC50s from the ER pathway assays. And then we compare those to the lowest effect levels from the guideline-like uterotrophic studies. You can see that when we do the comparison across a pretty broad range of chemicals, we actually do pretty well. In every case, we're within an order of magnitude. In most cases, the ranges of doses that we see in the high quality in vivo studies overlap with the ranges of doses that are predicted from the ER pathway assays. And then the last thing I'll talk about is accessing this data. So I already mentioned the EDSP dashboard as a great resource. Chris talked yesterday about the chemistry dashboard, which is a phenomenal resource. We have what I like to affectionately call our baby dashboard, and it's ICE. It's the Integrated Chemical Environment, not the mean agency that tries to keep people out of the country. And it is intended to be a resource for the entire community. So for test method developers, we have a number of well-vetted lists of reference chemicals, some of which I've already talked about today, others of which pertain to other toxicity endpoints. So any assay developer that wants to know, okay, how do I validate my assay or test it against a set of chemicals for which we know the activity, can come to our site and download these sets of reference chemicals. We also have all of our NICEATM data is housed in a computable, searchable format in the integrated chemical environment. The next thing that we'll be putting out within the next couple of months is a computational tools and workflows section where things like the IVIVE workflow can be downloaded and run on your computer. Eventually, we'll get to a point where we'll actually have a user interface where you can run them on the fly on the website. And this is work that's led by Shannon Bell, who's a fantastic member of our group and leads our computational toxicology work via the ILS NICEATM contract. Okay, so in summary, I've talked about a couple of different models that are relevant to endocrine disruptor screening that are actually being used for regulatory purposes right now, and some ways that we can contextualize the results of those models, both by quantifying the uncertainty around them and by extrapolating what they mean in terms of real-world exposures. And as always, this is a living, breathing project, and this work continues to evolve to include other really important endpoints and pathways like steroidogenesis and thyroid and things like that. So I'm just up here talking about this. There are a vast number of people who did the majority of this work, and in particular, Richard Judson and Kamel Mansouri, who's now at Cytovation, Eric Watt, who's now at Pfizer, Xiaoqing and Shannon in particular from ILS, and then many, many others from EPA and NIEHS. And I'll be happy to take any questions.